We wanted to try and model this, and we decided to use as a starting point this really elegant study by Jared Galloway and colleagues that was published, I think, earlier this year, maybe late last year, using SLIM. So basically came up with a model that started off with a marine population, which could then colonize these freshwater populations and run for 100,000 years, basically generating freshwater standing variation in that marine population. And then in Jared's original model, he had them colonizing another set of freshwater lakes to see whether just that original standing variation was enough to form freshwater adaptive populations. We've changed it slightly, so what we've got is at generation 100,000, we see a single colonization event into each of 25 lakes, in each case by 200 marine fish. And then we let those lakes evolve independently for 100,000 years. And because of the time constraints, I'll just really quickly run through one set of results. And so here we've got generation 0 in the isolation lake, and the allele frequencies more or less, well, they do resemble the same as the marine population at generation 100,000. And so that's why we see rising to high frequency, these marine alleles. And of course, they pretty much they rise to fixation quite quickly and they stay fixed. But after that, we start to see this gradual increase by generation 10,000 of freshwater alleles. So that's taken a long time though for that to occur. And just as a quick snapshot, I wonder if I can move this box a little bit, just out of the way. So parameters influencing chronology and tempo of adaptation, starting frequency, dominance, effect size, and recombination rate. Probably none of those are a surprise, but the recombination rate was actually less of an effect than we thought. So we changed it by an order of magnitude, and it wasn't a great, it wasn't a great explanatory factor of variation between which alleles rose to high frequency or not. But these simulations do suggest that it's actually quite a slow process, which is actually in contrast to some empirical studies which have actually found very rapid change to freshwater form. And perhaps it's all a little bit dependent upon things like migration rate and factors like that. So what we wanted to do next was, oops, I've pressed the button, nothing's happening. Okay, is actually compare the variation in freshwater and marine alleles in one of our ancient stickleback to see whether it actually matched up what we found in our SLIM simulations. And so in particular, do the variables that we've identified as being important for which freshwater alleles rise to high frequency first, does that correspond? Now that's actually work that we're still doing. We should have that finished in July, and hopefully we'll have something posted in the bioarchive soon, and we'll get an updated video posted at some point too. But what I want to highlight today is the variation across the genome, and this is basically ancestry in the ancient sample from the Ossavanet, conditioned on whether an allele, conditioned on the allele frequencies in top row, the Jones et al. Data set of 21 genomes from across the globe, and the second row conditioned on the local population. So whether it's alleles that are highly differentiated in the modern population. And what we can see, if we just take the broad overview, is that overall, the ancient sample still seems to be mostly marine, but with some really distinctive freshwater regions. So here in particular, large blocks of freshwater alleles. But perhaps the second most noticeable thing is that there's regions which are ancestry informative in the Jones et al. Data set, but they're not in the modern data, in the local data set, made up of the modern samples from the local lake and the local fjord. So here, for example, we can see this region here is clearly marine when we use the Jones et al. Background, but then when we use the allele frequencies from the modern samples, it doesn't really tell us anything. And we can see here the genotypes. So here's the Ossavanet Lake, and we can see that the Ossavanet Lake actually has marine alleles in this region here. So we can see why this is not ancestry informative. And I think really what the take-home message is that, well, first of all, these sediment cores from isolation lakes, they can be used as these evolutionary time series, so they can provide a time series data set of not only the genotype, but also the environmental change. So we can look at these concurrent changes. But in the absence of gene flow, we find stochastic elements of parallel evolution of freshwater sticklebacks. So there's all sorts of things that could be at play here. One is the reduced effective population size through time. Like I said, it could be this ticking clock where essentially selection becomes weaker through time and the alleles can't rise to high frequency. But also the starting allele frequencies might be a key factor in constraining parallel changes. And it could just be that if the lake stays completely isolated after the initial founding event and those alleles are not present in that colonizing population, then you don't have any standing variation for selection to act upon. You're essentially waiting for de novo mutations. So that there could be much more stochasticity than is perhaps you would expect from the current literature. In fact, there's just been a paper out in the past week by Fang et al. In Nature, Ecology and Evolution, which suggests there is this stochasticity or this reduced parallelism when you compare the Pacific to the Atlantic due to this founder effect and loss of genetic diversity during that process. And what we're suggesting is actually quite a similar process here, but on a lake-by-lake basis, so that you might end up with this variation in stickleback evolution because of these constraining factors. And this, of course, is highly collaborative work, and I should say this is part of Melanie Kirch's PhD study. So she's doing a PhD with Felicity in Tübingen, Felicity Jones, and Melanie was due to present another talk, another part of a PhD at the SMBE. So hence I'm presenting this today, but we will get Melanie to present this in an online video soon. Anders Romanset is the geologist from NGU who's been collecting all these cores, and it's been fantastic to work with him and hope to build up on this and join NGU for their 2 large-scale surveys they're about to do over the next couple of years. And then my long-term collaborator, Tom Gilbert, who has given me access to these ancient labs and really made the ancient DNA work possible. And of course, the funders, Marie Curie Actions and Bangor University for doing the co-fund scheme and allowing me to do this over the last 3 years. And of course, Sissel and Rebecca for organising first of all the SMBE symposium and then putting together this great online symposium. Given the sort of difficult circumstances, it's great to have this to fall back on. Thank you very much.